So first of all, I want to make sure that that everybody understands those are my opinions and not reflect the views of Nestle Purina Petcare. Those are my financial disclosure. I'm an employee of Texas A&M University. We do GI function testing and also microbiome testing on a fee-for-service basis. I'm in the Petcare endowed chair in microbiome research from Purina Petcare, and I also have a lot of speaking and consulting engagements with many different companies. So ultimately, what is the microbiome? I think we need to now put this in a clinical patient. I think we need to kind of a little bit rethink and take a step back and rethink physiology. In the past, we focused a lot on the host side, right? We looked at the epithelium, immune system, inflammation, and so on. But if you actually start thinking about physiology, this is actually the colon of a healthy cat. That's a true picture, and that's Dr. Chi Sung, she's one of my PhD students, did this really beautiful picture where you can see this is the cut to the colon of a healthy cat. And you can see here what we usually see as feces or intestinal content, it is actually loaded with a lot of bacteria. Nicely to see. So there's a lot of bacteria in the GI tract. We estimate somewhere between 40 to 100 trillion bacterial cells are in the lumen of the GI tract. Then we have this here in this cartoon, this mucus layer. And here, that's what you can see in black. This mucus layer is a very, very important safety, kind of like a part of the structure, because it really separates a lot of what happens inside the lumen of the GI tract and kind of buffers it from the epithelium. So this mucus layer is very important. And then with the epithelium, where we look at the epithelium with epithelial cells, where the absorption happens, and so on. And I think it's important to understand that this is all one unit, right? Often we kind of focus on the microbiome, we focus on the host side, but it's important to point out that both of them are interactive. We cannot really separate the host from the microbiome, and we can really separate the microbiome from the host. And what that means truly is as an example, we're going to see afterwards that antibiotics clearly induce dysbiosis. Yet not every single animal, actually majority of animals, not going to have any at the moment clinical signs, and most likely is because if we have all those other structures, like the mucus layer is intact, the epithelium is tight, and so on, the GI tract has a lot of capability to absorb those damages. But over time, if for many different reasons, acute injuries or like chronic injuries, chronic inflammation, these structures break down, then ultimately we're going to have changes in the epithelium, changes in the mucus layer, and that's ultimately going to interact with the microbiome. So it's going to cause some changes, and then ultimately, I think both together, microbiome changes and epithelium, is probably something that's going to kind of continue the cycle in the future. So again, important is that we cannot really manipulate the microbiome without understanding what is the secret of the rest and so on. And I think that's very, very important. We're going to see later on that especially once we have chronic GI disease, chronic mucosal changes, often the dysbiosis is persistent. That means we're not really treating dysbiosis by itself, we're modulating the environment in the guts, and that's going to be for us the longest, long-term, the best approach. The second thing is also that bacteria are everywhere. Dr. Paula Jarretta, who's going to be one of the next speakers, during her PhD, she did very nicely. So here, for example, you see the colon, the crypts in the colon of a healthy dog. And here on H&E, we don't really see much here when you look at deep side, but if you start silver staining, suddenly you see that in healthy dogs, completely healthy animals, you see bacterial structures appearing. And we then stain this with fluorescent stain, you can see that the crypts of the colon are really full of bacteria. And again, this is completely normal flora. And actually, Paula also showed nicely in this publication that in intestinal disease, in chronic intestinal disease, those bacteria that are here, they actually diminish, or they disappear, or they are reduced. So again, showing that being exposed to bacteria is completely normal. Bacteria are everywhere. And from a clinical point of view, if you look at a clinical case, it's important to appreciate or find the right balance, that how much, if I see bacteria on an immune slide or a histo slide, how much clinical relevance has this? So we kind of really have to put this into this balance. So what is ultimately a microbiome? I think when you look at from a big perspective, the microbiome in most mammals, at least in the large intestinal tract, is really consists of obligate anaerobic bacteria. So the majority of bacteria are strict anaerobes. Most of them are here like firmicutes, the Bacteroides. If you look at dietary changes, for example, diet is a very, very important factor as a substrate for the microbes. However, generally, the major phyla, the major conditions should not really change on diet. And you can see this very nicely in the study here, where dogs were fed different diets. One of them was a high protein, all meat raw diets. Then the dogs were transitioned to a high fiber, high insoluble fiber diet, and also high loss protein diets. The two latter ones were extruded diets. When you can really see the big picture, yes, there are small changes, but the overall conditions, like for example, the majority of bacteria remain strict anaerobes, and here proteobacteria, those are some of those facultative anaerobes that often, when they increase, that is a sign of dysbiosis. The same thing happens across many other mammalian species, like wolves, for example. Independent where they live, the majority of bacteria are strict anaerobes. So we have Bacteroides, Fusobacteria, and a small percentage are like E. coli. So in the past, we always thought E. coli is a major important bacterial group in the GI tract, and it's always there, but usually in a very small percentage because the majority of bacteria are strict anaerobes. And that's also important. Later, when we diagnose dysbiosis, we need to understand what actually E. coli means. So what is ultimately dysbiosis? And I think it's an emerging concept, but I think from a big picture is dysbiosis can be clearly defined as if there are clear changes in bacterial population, loss of diversity, loss of important normal taxa, potentially overgrowth of pathobionts. So we're going away a little bit with the term anthropathogens, because often some of those classical pathogens that we thought about, they are more or less an overgrowth bacteria. So it's not very clear-cut if they truly induce the disease, or they're more if some of the good bacteria, disappear, then we have an overgrowth of those pathobionts. And the second, I think, important definition of dysbiosis can be very clear if there's a change in function. If there's a microbial function that's very clear established, and this changes because of disease or antibiotics, it's clearly also a definition of dysbiosis. And the microbiota does a lot of different functions, but I think a few of them, some of the most abundant functions are the bile acid conversion, so conversion from primary to secondary bile acids. We know that the microbiota produces a lot of important compounds like short-chain fatty acids. And another very important pathway is the so-called tryptophan serotonin indole pathway, because that's also a very, highly abundant compounds in the lumen of the GI tract, and it's modulated by bacteria. And so you can see this nicely when we use antibiotics, for example. I think for the last few years, or since a few years, it's very well established now that antibiotics, especially broad spectrum antibiotics, can clearly induce negative shifts in the gut microbiome. So this is a study that was published in GBIM by Rachel Peeler. You can see here, that's the baseline in healthy dogs. We have this normal anaerobic flora. And then when we give metronidazole, after 7 days, you can see this huge bloom of Enterobacteriaceae or proteobacteria. So a lot of increase of other bacteria, so a very strong flip in the microbiome. When we stop antibiotics, so 14 days after stopping antibiotics, you can actually see that the microbiome reverts back. So it seems to kind of grow back. But if you're really very careful, you can actually see that those green areas, all those proteobacteria, E. coli, are still present in a high abundance here, even 1 month after giving antibiotics, when you really compare this to the baseline. So clearly, those shifts in the microbiome to antibiotic is a mimic, and we see these very similar shifts also in chronic GI disease. And ultimately, when those really major shifts, so you have a loss of those anaerobic flora, bacteria that are important functional markers, then obviously we're also going to have a shift in function. So the gut microbiome is considered a metabolic organ. We understand several pathways now. Some of them are very beneficial, like, for example, short-chain fatty acids, like bile acid conversion, so conversion from primary to secondary bile acids, but also some of the negative ones, like peak uremic toxins that are produced by bacteria. And if there's kidney disease, the kidney cannot really clear those uremic toxins. It accumulates and ultimately can cause further damage and so on. But important is that we need to understand, if you have a major shift in the gut microbiome, we're also going to have a shift in the gut microbiome, in the function. And one pathway that we really now probably very well is established is the bile acid pathway. So gut microbiota, so primary bile acids come from the liver, are converted by gut bacteria into so-called secondary bile acids. And in dogs and cats, so far, it appears that Clostridium hydrononis is the main bile acid converter in dogs and cats. So that means is those bile acids from the liver, primary bile acids, get converted by Clostridium hydrononis into secondary bile acids. So far, this is the main converter. We have not yet identified any other species that would do this function. That makes Clostridium hydrononis really one of the key bacterial taxa, key bacterial species, beneficial species in the gut microbiome of dogs and cats. And there's just an example is that those ratios, this conversion of primary to secondary bile acids is very important because now bile acids are well recognized as signaling molecules. So the proper ratio between primary and secondary bile acids in the GI tract, in the lumen or in fecal samples, it should be mostly secondary bile acids. In a normal gut microbiome, we have conversion. That means when you look at bile acids in feces, it should be mostly secondary bile acids. And you can see they really are agonists for many different receptors across many different organ systems, not just in the guts, but also in macrophages, in muscles, in fat tissue, and so on. And you can see nicely, there's a lot of data now is that this conversion is crucial because that really does a lot of physiological pathways. So this is a beautiful example, the bile acid pathway that shows you how the host and our gut microbiota are to some degree a part of our physiology because no other enzymes are available, host enzymes that really can do this. It's only bacteria. And this pathway seems to also be very conserved. So when you actually look at here in gray is the reference interval for healthy dogs that we have in our innings. And even if you look at very wild dogs, those are the New Guinea Highland dogs. There's samples that were collected. This is one of the most rarest dog breeds that is not domesticated yet. And you can see they also Clostridium hydrononis, and they also have this bile acid conversion. When you look at captive wolves, they're captive, but they are fed really raw meat diets. Again, they also have Clostridium hydrononis in the reference interval of dogs, and they also have this bile acid conversion. And we're now doing several studies in other animal species like tigers and cheetahs and so on. All of those animals have Clostridium hydrononis, and amazingly, they also have it almost at the same amount like we see in dogs and cats. Now, when you contrast this now in dogs, for example, here dogs on baseline, Clostridium hydrononis, the bile acid conversion, they receive metronidazole. You can really see how Clostridium hydrononis disappears. And also the bile acid conversion disappears. So the moment we have loss of Clostridium hydrononis, we really have also loss of bile acid conversion. And when we actually stop metronidazole, you can see that in some animals it comes back. Hydrononis, also bile acid conversion, but in some it stays persistent normal. So again, that's why antibiotics have to be realized that they have a long-lasting effect on some of the key bacterial species. And ultimately, in some animals, they don't recover, or it takes really weeks to months for them to come back to recover. But the bile acid conversion is really a conserved pathway, so we need this bacterial taxon, Clostridium hydrononis, because that is really the taxon that converts those bile acids from primary into secondary bile acids. And this has also been shown from many authors now. So for example, this is a study that looked at dogs with chronic enteropathy, and they all responded, or in this group, they focused on those dogs that responded truly to a hydrolase protein diet. And they found this very good association between improvement in clinical remission and also improvement in Clostridium hydrononis and improvement in bile acid conversion. So those dogs were induced in remission by hydrolase diets. They also saw that in the end, when there was improvement, there was a decrease in pathobionts. But importantly, this decrease was really associated because the microbiome slowly recovered. They saw an increase in Clostridium hydrononis and also an increase of secondary bile acids. Again, so this is very clear. So again, there's an improvement in the microbiome structure after dietary transmission. I also might point out now, and also come back to this later, that this is still a very slow recovery. Okay, so we discover now that especially in chronic enteropathy, this recovery is it takes much more time. There's association with this recovery, but in some animals, it actually still stays persistent. So ultimately, how can we really assess dysbiosis in clinical practice, right? And so this is really routinely offered commercial tests. So those are available to veterinarians. And in the end, it's bacterial culture. In some countries, in many countries, it's still very commonly offered as a marker of microbiome shifts or dysbiosis. And it could be also quantitative PCR. So for example, either for specific enteropathogens or pathobionts or the dysbiosis index. So how about bacterial culture? I told you before that culture is not really probably a very good tool. And the very simple reason is that most bacteria in the lumen of the GI tract are strict anaerobes. That means they're not really easily culturable with the routine techniques. So in those countries where culture is still offered, it can be a very useful tool if you're looking for very specific pathogens. If you have a suspicion of Salmonella or Campylobacter jejuni or other bacteria, then a culture is great because it really targeted. It allows us, once we grow this organism, to also do antibiotic accessibility testing. But it's important that culture by itself can only detect a very, very small percentage of those bacteria. And in this study, it has been shown that the authors, Melanie Werner, who's going to talk tomorrow for us about acute diarrhea, she has shown that they submitted feces to the 3 different commercial laboratories, and ultimately there was really no difference in the results between healthy dogs and dogs with chronic diarrhea. So culture could not distinguish them. There was a high level of interlaboratory variation. And importantly, because in some countries, there's this belief that hemolytic E. coli is a pathogen. And so in chronic GI cases, when culture is performed and it comes back positive for hemolytic E. coli, it's still sometimes suggested to treat them with antibiotics, but it's important that actually hemolytic E. coli are completely normal flora. And in this study, it was actually even more frequently cultured from healthy animals than from disease. So we need to be really careful in the overinterpretation of hemolytic E. coli as a pathogen. It's really normal flora, and there's really no association between healthy dogs and dogs with chronic diarrhea. So it's important, again, coming back to this concept, is that the gut microbiome is strict anaerobes. Now, we know there's a lot of different species. So in this study, for example, those are 12 dogs, but we have this study repeated many times, and other authors the same is when you look at really just the bacterial taxa that are present in every single healthy animal, there's really only maybe 25 to 35 to 40, depending how you define core microbiome. Does it have to be present in every single healthy dog or in 8 to 90%? That means there is not really that many bacterial taxa that are really present in every single healthy animal. Those other 1,000 species, they are kind of randomly. So yes, there are tons of other species, but the core bacteria, and likely those bacteria that are truly important for bacterial function, those are just actually quite limited. And so I'll give you an example that Clostridium rhamnosus, this Dysbiosis converter, if you do shotgun sequencing, it is truly present in every single healthy animal. And it's also quite abundant. It's roughly between 5 to 10% of the total population. Fecalibacterium are also very well-known bacterium that is a short-chain fatty acid producers, also present. So again, it's important is that we have several hundreds of thousands species present in every single animal, but the core microbiome, those species that are present in every single healthy animal, is actually quite limited. And so I would say somewhere between 25, 30 species and so on. And so that's why also when we started looking at those with targeted qPCR, we identified those bacterial groups always present in healthy animals, and often they are either reduced or increased in dogs that have either GI disease or have antibiotics and so on. And so when you target those key bacteria, so some of those core species that are present or should be present in every animal, we can kind of develop kind of a targeted assay that really can tell us how much the microbiome shifts. So this dysbiosis index, how it works is that we have qPCRs to retarget some of the beneficial bacteria that are known, like Fecalibacterium and especially Clostridium rhamnosus. And then also a few of them that are kind of overgrowth bacteria, like Streptococcus E. coli, Streptococcus, those are lactic acid bacteria. If there's some malabsorption, maldigestion, then they cannot really like to overgrow. And the more those bacteria go down and the more those go up, the more the so-called we can see a shift in the dysbiosis index. And the dysbiosis index shift tells us more or less how much the microbiome shifts away from normal. That most dogs should have been indexed below 0 or between 0 and 2 is this kind of this equivocal range. And the more the index shifts, the more you have loss of taxonomy, loss of diversity, and so on. And I'm going to give you later a few examples is that we discover now that depending on the underlying cause, if you have an increased dysbiosis index, that's usually in many cases often a sign of chronicity, of persistence. That means that's a marker of potentially already mucosal changes on the mucosal level. And again, coming back to Clostridium rhamnosus, it's a very important bacterium because we have several studies that really nicely shows that just the loss of Clostridium rhamnosus is already a major indicator of a major microbiome shift. So in this study, for example, you can see when we measure Clostridium rhamnosus by qPCR, if it's normal here in yellow versus if it's very low It is a bacterium that becomes very low, and you can see just looking at Clostridium by PCR, we can actually almost separate the gut microbiome into normal, abnormal. So this bacterium is so important because it is this bile acid conversion. And also show that we have, when it's less than 4 log of DNA, that is clearly associated with abnormal bile acid conversion. So Hieranonis is a key species, and this by itself, loss of Hieranonis really is associated with a microbiome shift that is not usually seen in normal. And we have this also validated in a recent publication where we collaborate with a huge list of experts in the area, that all of them had samples that are really well defined, because it's really crucial to define really well-defined phenotypes. Healthy dogs with acute diarrhea, chronic diarrhea, all of those animals were really worked up properly. So we looked at almost like 300 dogs with various phenotypes. And what we see, and it's a little bit of a complicated graph, but I think it's very important to show that. And that means is that we had samples with clinically healthy dogs, dogs with acute diarrhea, dogs with chronic enteropathy, non-GI disease, and dogs on antibiotics. And green means the index was below 0. Yellow means index below 0, but at least one taxon was of reference interval. So we call this a minor change. And you can see most of those animals really cluster in this area here. So we can really see that all of those dogs cluster together here. Reds are dogs that are between 0 and 2. They're kind of at this age of normal, they're on the side, but you can really see that those dogs are starting to cluster away a little bit from all the other dogs. So we can really separate those out. And then the higher the index, blue is everything about 2, but the higher the index, the more away from this normal those dogs are. So here are dogs with an index of 4, 5, and those here, very far away, are dogs with an index of almost 7, 8, 9. So you can appreciate it, that we kind of more or less have an estimate where a huge range of normal is. It's somewhere between around no, those between 0 and 2 at the edge of normal. That means they could already be abnormal, but they could be also just at the edge. But clearly, if we have an index of 4, 5, or even further away, so the higher the index, the further away it shifts from the normal range, that is really severe, severe changes. And it's important because a microbiome by itself cannot shift that much unless there's something really major in the environment. And as you can see, in most cases, it's really antibiotic usage, broad spectrum, that causes severe dysbiosis. And there's a subset of animals that have chronic enteropathy, have GI disease. But also this data confirms nicely that not every single dog with chronic GI disease has a dysbiosis, or at least detectable. Because those dogs that are here with normal index, they also are here. So even on sequencing, we cannot really find major differences. And that also means that when in the future, we can probably help stratifying us. There's a difference probably in those dogs that have dysbiosis and those don't have. And so with time, we're probably going to have another additional tool to help us stratifying in the different pathophysiologies. But ultimately is that the index really reflects very nicely and well, accurately, how much the microbiome shifts. So again, a very high index indicates severe, severe changes in the gut microbiome. And we also kind of, with time, we discovered that those phenotypes, for example, that dogs with chronic enteropathy, a subset of them, they usually have an increased dysbiosis index. Again, not all of them, but usually it's associated with a loss of Clostridium hieranonis. So the higher the index becomes, at some point, the microbiome shifts so much that we always have a loss of Clostridium hieranonis. But it's not a predictor how animals are going to respond. So dysbiosis is a part of chronic enteropathy, but it's not going to tell us really how they're going to respond. So again, we're going to talk about this later over the next 2 days, is that we're not really treating a specific marker, we're treating the whole environment in the GI tract. In acute diarrhea, I showed it before, and also here, is that it's a very transient dysbiosis. Most dogs have minor changes, so we detect a few changes, but very few dogs have an increased dysbiosis index and hieranonis. And so this is a very transient shift. But we also know that antibiotics induce dysbiosis, and also omeprazole. And Dr. Tolbert's going to talk about tomorrow the rational use of proton pump inhibitors. We see this also in cats. In cats, again, there's a feline index that also shows you that roughly 70 to 75%, there's 3 studies now from 3 different countries, show similar distribution. Roughly 75% of cats have an increased dysbiosis index. And it's really not necessarily related to the major symptoms. As you can see, also cats that don't have diarrhea, don't have vomiting, but just have those unspecific signs like hyporexia, weight loss, lethargy, they also can have dysbiosis. Dysbiosis reflects really, in those cases that are persistent, more changes in the gut environment. And if you put this all together, it's most likely truly, if they persist, it's really through those mucosal changes. So again, I told you about antibiotics, why we try to avoid them. So this is a study that has been done, again, from Rachel Piller in dogs that received high protein diets. when we have diet changes the substrate. Diet is super important, but diet, as also showed in the first slide, should not change, even on sequencing, should not change the gut microbiome really drastically. So we're going to see differences, but we don't really expect a major shift. And this study showed on sequencing and on the Dysbiosis index that actually most dogs stay in the normal range. But then when you use metronidazole, we have a huge increase in dysbiosis. And again, in many animals, it recovers, but in some, the recovery is very slow. And it's mostly Clostridium rhamnosus, as you can see nicely here. It's in a reference interval for dogs during dietary transition. We give metronidazole, we see a decrease. In some dogs, it recovers, but in some dogs, it doesn't. And those are also the dogs that don't recover in hyaluronosis. They also have abnormal bile acid conversions down the line. So it's a very individualized response to antibiotics. Okay. Omeprazole does also increase in the dysbiosis index, but importantly, it's a very more milder shift. Now we see they are kind of in this range 0 to 2, so they're the edge of normal. And when we stop omeprazole, it's actually really nicely reversible. So they come back within 2 weeks. Importantly, this is mostly through Streptococcus. We see this increase in Streptococcus species. We stop and it comes back. And it does not really change Clostridium rhamnosus. So that's the nice thing about it. We sometimes can see that different treatments, different dysbiosis pattern are different based on the source. So proton pump inhibitors make a transient dysbiosis. When we stop, it comes back, but it doesn't really shift. It doesn't have the same consequence, like, for example, in chronic enteropathy or in some antibiotics that lead to a loss of Clostridium rhamnosus. So why is this important now also for pathogens? Because I think we need to understand that the microbiome is important for regulating pathogens. I mentioned before that Clostridium rhamnosus and bile acid conversion is very crucial for reduction of pathobionts. Okay, and this is a study we did in young puppies. They were collected. We have samples that collected every few weeks, and you can see many puppies completely healthy. When they're very young, they have a quite abundance of Clostridioides difficile, but no clinical signs whatsoever. And then suddenly at week 7 to 9, this abundance drops and disappears. And the reason for that is because that's the time frame where the normal microbiome develops. So you can see week 7 to 9, we have an induction of Clostridium rhamnosus, so that the normal microbiome comes, the normal function starts. So we don't have bile acid conversion. So at those early, when you don't have hyaluronosis, no bile acid conversion, hyaluronosis comes in and we have bile acid conversion. It's very well known and also shown by the paper I showed in the beginning is that bile acids are important regulators and they actually reduce those pathobionts. And that's important. So this is not a reason to treat. This is more an indicator that microbiome is not normal. In puppies, in the very first weeks of life, that's normal. But once it starts, that disappears. And this is also important. We have a nice paper now from Melanie Werner again, where we looked at a huge population of dogs and show that the presence of Clostridioides difficile is truly highly, highly associated with dysbiosis and especially lack of Clostridium rhamnosus. So roughly 30% of dogs that we looked at in chronic enteropathy had Clostridioides difficile, but 90% of those dogs also had an increased dysbiosis index and no hyaluronosis. So what that means is that the presence of Clostridioides C. diff was really as a secondary overgrowth because we have dysbiosis, and dysbiosis pattern is a lack of Clostridium rhamnosus. And most importantly is that it didn't really change treatment response. Okay, so while many of those dogs were positive Clostridioides difficile, it did not affect how they responded to diets, because in dogs with chronic enteropathy, we don't really treating specific pathobionts. We trying to modulate the whole gut environment, and most dogs were food responsive, and it did not change if they were positive or negative Clostridium difficile. So again, the presence of some of those pathobionts, like Clostridium difficile, at least in dogs and cats, is clearly associated with low hyaluronosis, but it does not require changes in routine treatments. So again, like we're going to talk about the next 2 days, diet is going to be the main treatment in those chronic cases, because all of those chronic enteropathies, they associate with some change in the gut microbiome and to some degree overgrowth of some of those pathobionts. Same with Clostridium perfringens, very important. In acute diarrhea, yes, we have microbiome changes. They are usually kind of not as severe as we see in chronic GI disease. Usually there's no loss of Clostridium rhamnosus, and it's transient. And this is actually a very nice study from Andrea Reisinger from Munich. She presented last year at ACVIM. And you can see those are dogs with acute diarrhea, and many of them have an increase in Clostridium perfringens at baseline. But ultimately, there was no difference in clinical outcome between patients that were treated just symptomatically, or they received FMT or with antibiotics. Clinically, they all responded the same. And as you can see, also Clostridium perfringens decreased by itself, right, independent of outcomes. So again, there's an explanation why SPC, it's an indication of minor shifts in the microbiome, but again, it does not require specific treatment. We're going to treat them with diet, with other treatments in most cases, and we're going to talk about acute diarrhea treatment tomorrow with melanovirma. So important again, those pathogens like E. coli, Clostridium difficile, Clostridium perfringens, they're markers of dysbiosis or some change in the microbiome, but we don't have to treat them specifically. Important is to understand that antibiotics do also in cats. Actually, cats seem to be even more susceptible to antibiotics. This is actually a study we did. There's actually one cat from this study, but this is Minerva. She belongs to a pathologist, Jiao. And this study was meant to be to just follow up healthy animals over time. And you can see Minerva was pretty stable in this dysbiosis index over 8 months. She also had Tiranonis always in the reference interval. And then suddenly, because of some nasal or respiratory disease, she received 1 shot of antibiotics, cefepime. And you can see it caused change in the microbiome that ultimately even increases dysbiosis index and clear loss of Clostridium melanonis. And this change really persisted almost for 8 months follow-up. So again, we need to understand that antibiotics, even if there's no clinical symptoms, they clearly disrupt the gut microbiome, right? And that opens up all the potential for overgrowth of pathobionts and so on. The good news, though, is that at least in research studies, and that's the future we're going to try to figure out this, but in research, if it's an antibiotic-induced dysbiosis, so here's a study that is from Dr. Fred Gushon and his resident Jamie Hui from Louisiana State University, also presented as an abstract. If you have healthy cats and they all receive antibiotics, amoxicillin, clavulanate for 10 days, they all have an increased dysbiosis index. But you can actually see that with fecal transplants, if they get a fecal transplant right after the last day of antibiotics, we can bring the microbiome really back very quickly. There's also a few studies now looking at probiotics, so also giving probiotics. The recovery is quicker, probably not as quick as with FMT, but it's quicker. If we don't do anything, you can see in cats a very slow decrease. So those in black did not receive anything. And you can see in some cats, in 2 of those cats, even 8 weeks later, after antibiotics, they still had an altered microbiome compared to baseline. So again, but the importance of this, I think, is to understand when can we manipulate the gut microbiome and when we cannot. So in antibiotic-induced dysbiosis, where we assume that the rest of the gut structure is still normal and we have a dysbiosis, we can bring it back. It's very different, though, if we have this established chronic GI disease. Okay? And I think intestinal chronic enteropathy, we're realizing more that those changes accumulate over time. They probably are there already way before there is clinical signs, and it's probably a mixture of different things. So we're going to have a dysbiosis, we're going to have this breakdown, this mucus layer, we're going to have changes in the mucosal remodeling, shortening of villi, which potentially to malabsorption, to some degree, lack of barrier, and so on. And so what that means is that when we treat those chronic enteropathy, we cannot just treat the microbiome. That's not going to be sufficient because there's many other changes. The dysbiosis pattern is usually coming back. And we have those studies now that shows if we just treat the microbiome in chronic enteropathy, they're all going to relapse because, again, it's not just the dysbiosis by itself. The dysbiosis is related to many other changes in the gut environments. And that's why diet, this micromodulation, is always number one. It's always going to be the base of every treatment. And I think the future is going to show how many other combinations can we do with microbiome. In the end, I think the future shows that most likely it makes perfect sense to modulate many of those compounds as long as you understand what is always the basis of treatments. And this is a beautiful example also from Paula, who's going to talk later today. That was her PhD and shows you how those changes really are. So this is the villus in the ileum, and you can see how nicely and long it is, right? A healthy animal. And here in brown, that's this brush border. In this case, it's labeled for the bile acid transporters, which are absorbed in the small intestine. But it's also appreciative how superficial this brush border truly is. So there's a lot of absorption happening here, bile acids, other compounds, and so on. And then once chronic enteropathy, you can see that the structure changes too. There's a shortening, a blunting, and this brush border is much more reduced. And this is probably one of the major areas where we have a reduction of transporters, right? And probably this low-level malabsorption is happening. And this is ultimately also, sorry for the slide, again, associated with dysbiosis, right? Many of those animals have malabsorption that potentially could lead to dysbiosis and also clear function. But the important thing is that this is not really the extent of those changes varies between animals, and it's very difficult to predict how each animal is. But this is probably the reason that often even it's just to understand that that some of those changes are going to probably persist, right? And so many papers have shown now, if you have short-term follow-up of dogs with chronic enteropathy after a few weeks, many of them go into clinical remission, but major changes in the microbiome, but also metabolome, they are much, much slower. And in some animals, they actually persist probably for many months to years. So in this study, for example, this study used a completely different animal protein-free diet. So the protein source was not animal. In healthy dogs, again, we did not observe a major change in the gut microbiome. It was the same. In dogs with food-responsive enteropathy, they really went into remission. However, the microbiome only recovered partially. And again, most likely just for realizing is that those changes persist. And ultimately, it is probably something that just need to be aware that often with diet, we can put clinical remission or other treatments, but need to be aware that those changes still maybe persist. And this is also the future, I think, to understand is that once we see dysbiosis, and especially severe dysbiosis, that is often a sign of chronicity if it's not antibiotic-induced. Antibiotic-induced, I can bring it back. But if it persists, there's a beautiful study from Jennifer Scheidman, who is one of the pioneers of fecal transplantation, as an internist from Manhattan. She showed this in the very early studies, is that animals who have either no detectable dysbiosis or mild dysbiosis, they respond to FMT much better, also clinically, more long-term. But animals with severe dysbiosis, again, indicating there must be something severe in the gut environment, even after FMT don't really correct, or if they come, they relapse very quickly. And there's more and more now papers coming out there showing something similar, that in chronic GI disease cases, a presence of dysbiosis, the more severe, can be a prognostic indicator for long-term relapses. So again, we can clinically manage them, but we need to be aware that those are most likely, they're going to stay abnormal, at least in those markers, and relapses are very, very frequent. So to summarize, I think it's important to understand what is the gut microbiome. It's mostly alblicate anaerobic bacteria. That's the majority. Proteobacteria coli are the minority. So that's why bacterial culture is not very useful if you really want to assess microbiome shifts. Microbial culture can be useful for very specific pathogens, okay? Pathobionts like E. coli, Clostridium perfringens, they increase in dysbiosis, many times in acute, but they're self-limiting. That means they indicate there's some disturbance, but we don't really have to treat it specifically. That's important. Clostridium difficile, or Clostridioides difficile, is definitely associated with dysbiosis, increased dysbiosis index, and especially lack of phenonosis, because it's really controlled by bile acid conversion. This is almost exclusively seen in chronic enteropathy antibiotics. But again, the studies, at least in dogs and cats, also suggest that we still doesn't change the treatment. We're still going to start with dietary and other, subsequential treatment as we do for chronic enteropathy. The index itself accurately reflects microbiome shifts, so a more increased dysbiosis index reflects major shifts, but it's really important to understand what is the reason for that. Is it broad-spectrum antibiotics? We know that broad-spectrum antibiotics can induce microbiome shifts. Usually they recover, or at least they're not going to be pronounced, they're not going to stay long-term pronouncedly high, okay? But in some animals, we're still going to see an index of 2, 3 long-term after antibiotic uses. And the other reason is chronic GI disease, this mucosal remodeling. And if we use fecal transplantation, we understand why is that. In antibiotic-induced dysbiosis, we can bring the microbiome back very quickly. However, in chronic GI disease, if it persists, and it's going to persist in the majority of cases, that really suggests chronic GI dysfunction that's going to probably require long-term management in those animals, multimodal management with different things. So with this, I would love to acknowledge all collaborators, a big team in my lab, in the GI lab. We have a really great lab now that we have pathologists and internists, and it's really nice. And then a lot of collaborators where we get a lot of samples from that really help us to really understand all those perspectives from acute to chronic GI disease and so on, and the granting agencies that sometimes fund our work. So with this, I'm happy to end.